Hello everyone. Today is a special day, so we don't have any special topic for today, just answering your questions. So you see what we are talking about. So actually we are talking about TRANSFAC, and TRANSFAC is our gold standard of gene regulation, as we call it, and it's quite old, I would say, but we are maintaining it for a long time. It's more than 35 years now when it was created, and now it's one of the biggest database related to gene regulation. And we have, if you read about TRANSFAC, you have TRANSFAC of different packages. We call it TRANSFAC Basic. So the one which you may know before. TRANSFAC Omics is something new. What we have now is actually allowing you to analyze your omics data. So all kind of omics, including transcriptomics, genomics data, like mutations in the genes. You can analyze your proteomics data, or very important to analyze epigenomics data when you have, for instance, you have toxic or gene or ChIP-seq data, and you know where transcription factors are binding or which regions of genomes are open and important for you, or you know which modifications of the chromatin are done in particular regions, and if it's actively changed or mutilated, you can give those regions to our tools and analyze them. And what exactly you're analyzing, we'll talk a bit today. And there are two other options which maybe you don't know yet, but it's very important. Basically, transcription factors are just end points of the complex regulatory machinery in the cells. And once we know which transcription factors are important for you and important for a given pathology or for a given experiment, then the actual question is whether there are some signal transduction pathways where these transcription factors are regulated. And these tools, which we have in this master regulatory, the TRANSFAC master regulators package, include so-called TransPath database, where we collect information about pathways and huge network about protein-protein interactions and reactions, signal transduction reactions, very detailed. And that allows us to find master regulators and to find potential drug targets. And of course, we have this full package, which include everything, and also a very special tool called Genome Enhancer. And I think there were some questions about this, and I will touch this as well. So these are our, of course, what we offer to you. But you, of course, know most is TRANSFAC database as it is, which is called TRANSFAC Online. And it's about transcription factors, about their sites in genome where those factors are binding. And very important part of TRANSFAC is a collection of motifs, we call it, or which are encoded in a form of positional weight matrices. And we have a biggest collection of those matrices for various species. And those transcription factors, we made a very good, attempt and a lot of efforts to classify them and to have a kind of, anatomy of all transcription factors in the human genome. So that's all included in TRANSFAC database. Okay, so we'll see a bit more of those kind of questions, but before you put anything in Q&A, let me go through my list of questions. And actually, the questions are very much, different. They concerning very philosophical questions like what is transcription factor, and towards very simple questions or very detailed questions, how to use a particular tool. So I have collected them, which we got in meantime and before the in the time before and after different coffee breaks. So the first question is very often, people asking, which transcription factors are regulating my genes? And this question is really a tricky one, in fact, but it's very simple, looking very simple. Which transcription factors are regulating my genes? But let's approach this question step by step. First, we can ask, reformulate these questions. Like I have my gene, and which are data available in TRANSFAC, published perhaps, or maybe if we collect it from some unpublished data, but from high throughput data or from other types of data, if there are some data showing that this gene is regulated by a particular transcription factors. So just checking what knowledge is there in the database. That's possible. So let's do that. Let's log in our database first, which has basically all subscriptions to our products. You see, we have here TRANSFAC online, and we have TRANSFAC, TransPass, and Human PSD. This is what I call the full package. We have here Genome Enhancer. We have here, this geneXplain platform. We will see today a bit more details. And there are other databases like Brandar and TransPass I mentioned already. So that's you might also interested to look at. But today we first we'll have a look at TRANSFAC. Actually, let's click on TRANSFAC. The question was, which transcription factors are regulating my gene? So, okay, let's type the gene, for instance, BRCA1, which is a very famous gene. And you see here a number of, different types of results. We found 50 genes or proteins with such a name for different species. Here you see they are listed for human, for rat, for mouse, and for other species, what we have in the database. We have also, matrices and other information, but let's dive into information about this gene. And of course, in the gene entry, there is a lot of information about this gene. And of course, you can read all those information about what kind of gene ontology it belongs to, what mutations are known for this gene, which tissues it is expressed in, and so on. But today we're just interested in one thing, basically, which transcription factors are regulating my gene. And to do that, we here just directly in this search interface, what you can do, you can simply select this gene like this and ask binding factors for the gene. Very simple, and click search. And with that, you will find which transcription factors actually are regulating my gene. And among the transcription factors, you see, binding factors like GABA-P alpha, with GABA-P beta, like CREP, another CREP is RTF1, the complex, the transcription factors acting not just individual proteins or peptides, they're acting as complexes, they're acting at heterodimers and so forth. So we have all this information here in the database, CMYC, CJUN, E2F1. So we have all this a list of transcription factors which are regulating my gene, BRCA1, right? And quite many of them. Actually, we can even go further, selecting now this gene, which encode for a particular transcription factor, and ask again this question. But if you go back, and there are a number of other things here. For instance, we can ask whether which transcription factors are regulating my genes or binding to the regulatory regions of my genes, which are known from ChIP-seq data. So we're collecting ChIP-seq information in our database, and the same thing, we can ask the same question. So which transcription factors are binding to regulatory regions of my gene, which include the promoter of that gene, the whole body of the gene, and also 3' end. So actually the whole area which can contain regulatory regions for the gene. And we check which peaks from different ChIP-seq experiments were actually detected from various papers, from ENCODE project, from other papers, and then we document this in the database, and now you can see which transcription factor and from which ChIP-seq data are regulating my BRCA1 gene. So you can see this list. You can actually export this list. You see how many of those transcription factor, like 238, and you can save this result. You can export this result as a table for your further analysis. All right. So, but that's only half of the answer. These are transcription factors which we know from the literature, from the ChIP-seq experiments, which can either regulate my gene or just simply binding to regulatory regions of the gene without knowing, in fact, sometimes whether they are regulating this gene or not. They're just binding there. Maybe they're regulating some other genes which are nearby, but from ChIP-seq experiments, you just know they are binding. So that's a good hypothesis to assume that these factors are also regulating my gene. But actually, we know that this information is far not complete. So we, of course, do in our, very extensive efforts in TRANSFAC annotation to collect as much as possible available information from literature when people were doing experiments and confirming that this factor is binding to that site. And then it's regulating these genes. But these experiments are very expensive. They're actually difficult to do. And far not all transcription factors are actually known, which are regulating my gene in different situations. That's why it's important to actually predict which transcription factors are potentially may regulate my gene. And for that, we have a tool called Match Suite. It's a collection of tools, actually, because that's why it's called Suite. So far, it's applied to human, mouse, and rat data only. So you can put there a gene name from these organisms. In future, actually, pretty soon in this release, we are extending this list to other model organisms. But right now, it's human, mouse, and rat only. And then if you click on that tool, it has a very simple interface. You have some user guide how to do. Then you click on Start Analysis. And in case if you are analyzing human data, you have this, two tools. One tool is looking for a single gene analysis, and another for analysis of gene set. And there is a principal difference in these two approaches. We can discuss it. I think I have some questions about gene set analysis, but now we are looking for just one gene. So you click on Next, and now you put here the same name, for instance, BRCA1. You click Next. And of course, this tool will have to be kind of focused. It will take the whole library of matrices of motifs from TRANSFAC, and it will apply all of them. There are quite many matrices. We have, in fact, for more than 10,000 matrices now in TRANSFAC. But for vertebrates, it's around 8,000. So it's quite many matrices. And if we apply all this motif search for the whole region of gene, we may get a lot of sites. So that's why we usually want to focus on most important areas, regulatory areas of that gene. And here what you see is you have your gene, and you can select the tissue of your interest. And that's very important because, as you know, in different tissues and different cell types, there are different transcription factors acting, although they may be from the same family of factors. But some members of that family they expressed in deposit tissue. Other families of that, other members from the same family of factors may be, working in B cells. And to understand which factor is actually regulating my gene is important to understand which tissue of your interest. If these are like family members, and they may be expressing different tissues, but there are some very specific factors which are expressed only in one tissue, and they are very specific. There are no other family members which are expressed in other tissues from that particular family. And then also we know that in our database, and if you select a tissue, we apply a special filter, and we filter only those motifs, those positional weight matrices, which are connected to transcription factors which are expressed in that tissue. It is with a special specificity. There, of course, expression of a factor may be everywhere, and it's not enough for a factor to be expressed. You have to be activated, so the factor has to be active in a particular tissue. But here we pay attention to specific factors. So that's why when you select a tissue, then you may get a different result in one tissue than in another tissue. You can also go without selection. In such a case, you will basically apply all the list of matrices and consider basically the full potential of that promoter area to regulate the gene. Okay, and you can also check which tissues actually this gene is expressed, and this information is taken from various sources. Actually, our main source is a protein atlas, which gives information about expression of the gene in various tissues. And for BRCA1, you see it's expressed in very many different tissues, and but there are some tissues where it's minimally expressed. So as usual, testis is very, very highly expressed. There are a lot of factors which are expressed in that tissue. So you can look at this picture and select your tissue of interest also from a point of view of expression of the gene itself. So if the gene is really a very specific tissue-specific gene. If not, depending on your experiment. All right, so when you select a tissue, the next step is actually to select the promoter area of your interest. And that's by default, it's from -500 to +100 around transcription start site. And it's kind of a very core area of the gene regulation. But you, of course, know that in many cases, actually, regulation of gene is done by quite distal areas, and then you can customize your region to up to, I think, what was that? Up to 2,500 upstream and up to 1,000 downstream. And also you can select which promoters you, in fact, you are interested. One gene can have several promoters, as you know. And some promoters are very special, again, for a tissue, and that also considers when you select a tissue, for instance, let's select testes, right? And then the system knows which promoter is testis specific for that particular gene, for BRCA1, and will take the start site, the transcription start site from that, which is known to be specific for that tissue. And this information is taken from Phantom database, actually. And you can take this, this promoter. But also in one tissue, there may be several promoters. And you may select the best supported promoter, which means that promoter which has a maximum number of, transcripts, which is starting from this TSS. You can take also most proximal start, which makes sense quite often, because then this is the longest transcript which you're considering. And then there is a special regulatory machinery associated with a long transcript, perhaps. But okay, by default, it's best supported. And there is another point, which is called profile optimization. And what is a profile? There's actually one question. We'll talk about that a bit, maybe in details, but that's what we mean is, for instance, I select immune response or inflammatory response together. And what happens then? Then among those transcription factors, which we are now interested to know which transcription factors are regulating my gene, I can select a subset of them which are involved in, for instance, immune response. If I am really interested in the immune response only. Or I don't select anything. In such a case, all transcription factors are considered. And then you launch your analysis. So that's one step. And then you simply wait for the results. So we will have a look at the result section to see what we can get there. Okay, so it's preparing now the list of answers. You see this BRCA1 is running now. It's in progress. But we can have a look on what I have done before. This was already analyzed. We can simply click on view results and see what kind of information you can get there. So again, to repeat, initially the question was to check what is known information, which is documented in TRANSFAC. But now we asking a question, what can be a prediction? And the reason is that actually not everything is yet known, far from being complete. So our estimates actually telling that it's not more than 1% of binding sites which are known so far in the whole genome, and 99% are still unknown, or which you can predict. All right, so what we see here is actually these are three windows. And here you see this BRCA1 gene in the main promoter. And in fact, what this tool is doing is not only analyzing the promoter, but actually it's taking also enhancers. And it looks for those enhancers which are known for the selected tissue. And then it analyzes promoter and enhancer separately. And here you see this list of transcription factors, which the system found may regulate my gene. And these are 40 transcription factors. And here you see the view, basically the factor name, the name of that particular motif, so positional weight matrix. And then we have here a number of columns. First column is actually showing you the overrepresentation or enrichment of that motif in that one promoter. So what means enrichment? Actually, and in fact, it's a p-value of that enrichment. That's quite a tricky point, because we have just one sequence here, one promoter, and we have to estimate enrichment of that site in that one promoter. What happens if there's just one site for this factor? Whether it's enriched or not, we don't know. So to estimate that enrichment, what we do, we actually not just count the binding sites which are above a particular threshold, but we consider the whole profile, how this affinity of that matrix of that motif is changing across the promoter. And then if this affinity is, affinity profile is higher than usual, so higher than in random chance, we can estimate the p-value that this affinity to that factor is so different compared to the random sequences. And this affinity is accumulative. It's not just because of one site, it can be accumulative across the whole promoter, because for the affinity, not just one site may be playing a role, but other kind of degenerative motifs which are around that site can also contribute to the binding affinity of that factor. And we can estimate this p-value of that affinity. And this is minus logarithm of this affinity p-value, and you see it's 18, which is very, very significant. And we can, here we see also, well, in this case, in fact, what we did, we analyzed it not in the testes, but in in B cells in that particular example. And you see that this factor is actually expressed very well in B cells. That's why this factor is now put at ranked quite high in this table, because the affinity is pretty good, and also the expression of that factor in this tissue or type of a cell is very high. Other second factor, this one, also has a quite high affinity, and in addition to quite good expression, it has also this nice thing, which tells you that it's not only just highly expressed in that tissue, but it's also specifically expressed in that tissue. So 2 out of 62 means these B cells, in respect to the expression of that factor, is on position 2 if you just sort all expressions of this factor in all 62 tissues, the B cells and the number 2. So it's this factor is very specific in that tissue. So which means these are 2 levels of, 2 evidence, kind of levels of evidence, which tells you from one side it's this motif is very enriched, and on the other side it's also the factor is quite specific for this type of cells. You can see here also a detailed map of those sites. So if you see here the promoter, so this is a promoter of BRCA gene, BRCA1 gene, and this is a start of the BRCA1 gene, so the first exon. And each arrow here is predicted binding site for those factors. And if you click on each of that binding site, you see the consensus, the logo plot of that motif. You see the score of that site, and the highest score is 1.0, and if it's 0.88, it's pretty high. And there is a core score, the score of the core part of that site, which also very important for the binding. So there are other things here which I can detailize maybe in other talks, and you can also see. We had coffee breaks dedicated to the match suite especially, and you see also there are many parameters which you can vary here to understand your gene regulation. Okay, so you see it's just one question, half an hour. So I will try to be a bit shorter for the next questions. Okay, let me see. So the question from online, so from your list, are there modified forms of transcription factors in TRANSFAC? Yes, there are. So then basically what you have to do is to have the full package, or at least the package with a master regulator. And the reason is the modified forms of transcription factors are very important for the whole network. To understand the whole network, and then let me see. Maybe I just don't see them there. So this now I open the full package, and here you have also the pathways, not only information about the transcription factor itself, but you have also pathways, and here you might have the modified forms. I know that for sure there are number of modified forms for transcription factors. You see here, actually, there are more information in the entry. Right, so features. Yes, here you see the post-translational modifications of a transcription factor, and we have this map and all the kind of phosphorylated forms of a transcription factor. And they are not simply listed, they are in fact provided as an entry in TransPath database. So each individual modified form of a protein or complex of protein or any unit, we call it any molecule in a cell, which have a function, a particular function. For instance, the p53 modified phosphorylated in these 2 positions may have a special function. It is involved in a particular reactions. This positions are phosphorylated by a particular reaction. So you see here detail about that reaction. So taking from a particular, biochemical paper where there was one cell line and a very specific method used to understand how this P53 is getting these numerous phosphorylation points. And then after that, it becomes active in a special way. So that part is present in TransPath database. And to get information for that, you need access to the package which include TransPath, which is actually TransFac. It's called TransFac Master Regulator package or the TransFac full package. All right, so that's to this. All right, second question. Are there pathway upstream of transcription factors included in TransFac or just in the full package? Right. So basically, I answered partially this question. As you understand, those reactions, the whole network of reactions are stored in TransPath, and then to get access to this data, you have to have the package which include TransPath. But TransPath actually have not only all the kind of reactions, but it has also connection to pathways. Like when we typed transcription P53, you see there it also shows a number of pathways, and the pathway means actually it's kind of a canonical representation of a collection of reactions, which happens in a cell in a standard way. Of course, that's a very artificial construct, in fact, the pathway, but it also comprises a particular biological meaning. So the pathways which are canonical, if you can have a look, for instance, Aurora B cell cycle regulation pathway. This pathway, you see, this pathway is quite big. It represented in a number of reactions. You see, it's really detailized, and that's a difference to many representations of pathways you may find in other resources where it's just a picture or maybe it's just a graph. Here, each individual reaction is actually very described in very many details. But this is still a collection of reactions. And this pathway is actually composed on sub-pathways, so we call them chains, smaller connections, smaller sets of reactions which are following one after the another. And altogether, they actually can be seen as a graph. I think we can do it like here. So if you click here, I select the pathway and click on PathFinder. So you will see the whole picture of that pathway. And those pathways play a very important role in our analysis. For instance, if you have a set of genes and you want to make an enrichment, pathway enrichment analysis, then this enrichment is done by using these detailed pathways from TransPath, and you see it's the components of that pathway are not just individual proteins, but protein complexes and modified forms of those proteins, like here, or modified inside the complexes. So that's these are also quite a unique feature of our package, TransFac Master Regulator package, which contains such number of maps. All right, so that's about pathways. Next question. Can you search for specific microRNAs in TransFac? Yes, you can. Let's have a look. In TransFac, we actually we collect information about microRNAs from literature. And I wouldn't say it's covering all possible information about microRNAs. We do our best efforts to collect this information as much as possible. And so it basically, gene regulation is, traditionally And actually, in the major component of regulating gene is transcription factors on the level of transcription, but microRNAs are playing a role in regulating gene expression, mainly on translational, post-translational, I would say pre-translation level, and also known also on transcriptional level. MicroRNAs can play a role, and you can search for instance, here we have example of miRNA 155. So you simply type this like this. Let me see if we can get information about that. Yes, so you see this microRNA in human, in mouse, in rat, in other organisms. So again, you can click on that entry and you read information about this microRNA. And we actually try to collect information. We are primarily we collect information about experimentally proven facts, not just predictions of targets. And you see here information about this microRNA and in which biological processes it play a role. Very important, actually, and that's quite unique for TRANSFAC. microRNA are encoded by a gene, right? And this gene is controlled by normal polymerase II. So expression of microRNAs are controlled by transcription factors. And we have here information about which transcription factors are actually regulating expression of the microRNA. And that's very important, because when you analyze your microRNA arrays, or you are doing it by RNA sequencing, you are of course interested in expression of microRNA. You know which microRNA are differentially expressed and which are not change their expression if you compare different cell types, or if you do some treatment or some other types of experiment. And the standard way of analysis, which you can find in many other tools, you actually go downstream. You call it downstream. So you take a microRNA and you ask a question, which targets this microRNA has? And that's very important information. We have it here in TRANSFAC. But very specific type of analysis is asking a question, what are the controlling mechanism which controls this expression of that microRNA? We call it upstream analysis. We go upstream and we go to check which promoter actually involved in regulating expression of microRNA. And that's not an easy question, because some microRNAs, they have their own promoters. So, but many of them are just part of a kind of waste of other genes. So they are located in introns of other genes, and the expression of those microRNAs are depending very much on the expression of that gene. So basically, you have to know which promoter, in fact, is regulating expression of microRNA. So all this information is collected in TRANSFAC. And so this is also coming from ChIP-seq experiments. And then we have features of that microRNA, and we have, of course, a list of targets of that microRNA in TRANSFAC. All right. Okay, another question. The question is there a TRANSFAC module included in geneXplain platform? No. If you have a subscription to geneXplain platform, and let me just quickly show what probably you don't know, some of you don't know what does it mean, a geneXplain platform. Basically, if you don't have your account, you are not, you don't have a license for TRANSFAC, you still can get something to try yourself for free. So if you click here on register, you see here there is a registration for a free platform account. So you get a license key automatically, you put your name, and you get an account in a platform. And the platform, actually, it's here. You see geneXplain platform. Of course, since I have here, my account has a subscription to TRANSFAC, then my platform account has access to TRANSFAC. So here in the platform, and this is platform interface, and the platform is a collection of tools, actually platform, where we put a lot of tools to analyze different omics data. And here in the list of databases, you see I have a TRANSFAC database of different releases. I have TransPath database. I have human PSD database. I have all subscription to those databases, and they are connected to this tool. And then, which means if I analyze my RNA-seq data, for instance, and I want to discover different, analyze regulatory regions, for instance, identify enrichment of the transcription factor binding sites in promoter, then I have this option using TRANSFAC. But if you just register for free account of geneXplain platform, you get everything here. But you will not have here the TRANSFAC database, and your pipeline, which uses TRANSFAC databases, gets inactive. So as soon as you license TRANSFAC, you immediately get this databases linked to this account, or you create a new account for that. And then basically everything becomes active then in your account. All right. Okay, let me read further your questions. Thank you very much. What kind of information is covered in the mutation section of the result page when a specific TF is searched? Is it just specific to disease conditions, somatic mutations, germline mutations? Very good question. Thank you very much. And you have very good eyes to look at when you search here in the locus. Actually, when we was this, for instance, transcription factor entry. So this was an entry of transcription factor of P53. And so the question was about the very beginning of that entry. So here we have these mutations. And as you can see, these mutations are actually reflecting mutations in human gene mutation database, HGMD, which we don't have in our portfolio. But if you click on this, you can get actually QIAGEN page where you can subscribe to HGMD. So here what we have, in fact, is just number of mutations which are reported in HGMD. And NGMD, concerning your question, whether it's germline mutations or somatic mutations, they are focused on germline mutations. As far as I know, for a long time they were not actually extending their database to somatic mutations. Maybe they're changing their policy. I don't know for sure, but originally it's a germline, and those mutations which are related to a particular pathology or very specific phenotypes. So and again, you have to differentiate this from just variations or SNPs, which you have, for instance, in human in SNP databases like dbSNP, where all kind of SNPs which you observe in human populations are recorded. Here, the focus is on the real mutations which are causing something. And there are not so many, like all SNPs, you have millions of SNPs. Here you have thousands of mutations. But these mutations can actually affect encoding genes, and they change proteins, and that's a known fact. So the protein become either changing their activities, or they gain some function, or they simply lost function. So basically, those proteins are not functional any longer, like it happens in many tumors. But some of those mutations can affect binding sites of transcription factors. And in fact, we are preparing now a database called Transmute, which we hopefully release soon. And in that database, we actually analyze how these human HGMD mutations are changing the transcription factor binding sites for our transcription factors. And that will be very interesting information for you, perhaps, if you are interested in that topic. All right. So another question. Can you study the transcription regulation of any gene, for instance, NRF2 and its binding patterns in any conditions? Okay. Of any gene, yes, right. So exactly, that's what I was a bit touching, but let me make it a bit more clear. So let's just simply go and search for this gene. So NRF2. That's quite a well-known gene, actually. It's also a transcription factor. And it makes a little bit, tricky sometimes to answer such questions, because this is a gene which is encoding transcription factor. And this transcription factor regulates activity of other genes. But this gene is also regulated by some set of transcription factors. And the question was whether we can study this gene irrespectively from any tissue or conditions, just the full potential of that of regulation of that gene. Yes, just an entry of this gene. For instance, here when we look at which sites are there in the regulated regions of that gene. So we have here, you see, different sites, and each of that site was detected in a particular experiment. For instance, this is a RunX1 binding site in this very distal region regulating this gene. And if you look at the more detailed information about this particular binding of that factor, you can see that it was done in leukemia cell line with erythroid megakaryotic characteristic. And also polymorphonuclear leukocytes. It's very, a very strange kind of cells. And the experiment which was done with this type of experiment. But most important, related to a question, is basically which cell types or which conditions was studied when these researchers found this binding site. And it was, in this case, leukemic cell line. In other cases, for another site, for instance, here, we have, again, the same site, probably from the same paper, but this site is different, I guess. So that's another factor. And it's probably studied in a different experiment. So you see, it's embryonic kidney cell line. So it's a completely different tissue, different type of cell lines in a sense. This picture, which you see here, is an accumulation, is a summary of different potential this gene has in different conditions. Very important. It's actually a very important question to understand that when you analyze this gene promoter using, for instance, match tool. Let me find where I have my match tool. If I didn't find it, here it is. We select a particular condition. So in that respect, for instance, if here in this browser, you can select here experimentally validated binding sites. So basically, it will show here on the same map those binding sites which are known from TRANSFAC. You see these two ETF binding sites are known in TRANSFAC, but that doesn't mean necessarily. You see, there is no ETF binding site predicted here because we selected a particular condition. In that particular condition, ETF binding site may be not relevant here, although it's present in TRANSFAC database, and if you would select a different condition, we may find here this binding site. And of course, if you don't want to do a particular selection of a condition, let me maybe select the previous list. So then you select no conditions at all, and then it will, in respectively of condition, it will search for the whole potential. Still, in the match suite tool, we try to filter those binding sites from a point of view of affinity criteria. So before the searching for binding sites in your promoter, we estimate whether this motif has a potential to bind to this promoter from a point of view of affinity criteria. So we are actually filtering a set of motifs, and that's why the final output of the binding sites is pretty selective. So we have a kind of reasonable number of transcription factors and binding sites which are found in that promoter. If you want to understand completely, without filtering, without any prior conditions or prior knowledge, you have this possibility, of course, and that's this you can do in geneXplain platform directly. Let me show this to you, and this will be probably the end of my session today. Let me go to, I have too many questions, yes, let me open geneXplain platform. And in the geneXplain platform, as I said, we have a collection of original tools, which simply building blocks of these more complex pipelines. Like much of it is, in fact, a pipeline which applies a particular filtering of motifs and then other cutoffs for tissue specificity and so forth. And then you get a reasonable output based on our expert knowledge. But if you want to simply get the full potential of that regulatory region, you can simply apply all motifs from your tool, and for that you have here all the methods. Yeah, let me see if it's now acting. I guess probably you're in parallel. You started working with this tool as well, so that's why it's getting a little bit slow. Yeah, you see here we have these site analysis tools. And here we have a very simple tool called site search on gene set. And in fact, the gene set could be even just one gene. And then this gene is taken, and then you apply the whole library of matrices to analyze your, let me open it. To analyze your promoter of that gene. We open it twice, it's here. And as you can see here, you can put this list of genes, which can be just simply one gene. Let me select this gene. I want to show you step by step. This is important. You want to simply find your full potential of the promoter of that gene. I want to actually import one gene. Let's take, for instance, this P53. So if you have this gene in some extra table, or you can download it from somewhere, this is also possible, but you can simply type it as in this window. So it's actually from human. So now we'll have this gene. But actually, to know exactly what place in genome you are talking about, you have to actually convert it to ensemble ID. Why? Because just gene symbol sometimes is not taking you fully, understanding where exactly in genome. Some genes have several copies in genome, right? And they would have then separate different ensemble IDs. So in fact, what we first need to do, we have to convert this gene symbol into ensemble ID. And then our tool will know exactly which place of genome we are interested. So you simply drag and drop it here. You specify what kind of conversion you want to do, the input type, output type. So gene symbol is converted to gene ensemble. Okay, we are converting it. The same tool is actually converting the full list of genes. If you have, I don't know, 1,000 genes, or even a table with genes is with some values, for instance, expression values. This can be also, used, analyzed, and converted using this tool. And now we have this site search, and we have this gene. We put it here, and here you have we call it yes set. It means yes and no. Very simple to understand. So the yes set is your foreground, where you want to search for your binding sites, and the no set is your background. And that's very important, especially if you analyze sets of genes, and then you want to compare, and you want to find enrichment of particular motifs. You have to compare these promoters of your genes with promoters of other genes. For instance, promoters of housekeeping genes, or promoters of genes which didn't change expression. But again, here we want to analyze without any prior knowledge, so we don't need actually to have here no set, just one gene. Then you need to specify promoter, so which is minus 1,000, for instance, or is 1,000 nucleotides and plus 100 around transcription start site. And here is the list of motifs which you want to analyze. And by default, it's called non-redundant profile. It's a very small profile of motifs which we select for which class of factor one motif, just for a quick search. But in fact, if you want to apply all motifs, then you have to select here vertebrate human P001, for instance, this profile, which means all vertebrate motifs which are with optimized cutoffs, and I will talk about these cutoffs if you like, because just having a motif is one thing, but you have to set up a cutoff, a particular threshold after which you will call this as a binding site and not call as a binding site. So here we will select the non-redundant, and min sum means, again, it's around we are talking about cutoff here. Min sum is quite optimum cutoffs which balancing between false negatives and false positives, and it's reasonable to use. And then you simply run it for one set of one gene. It must be quite quick. Okay, cutoff optimization, no set. Right. So actually, by default, we have here optimized cutoffs, and that's actually good that it gives me this warning. You need, if you want to find enrichment and you apply your null set, then you can actually click here to optimize cutoffs. So which means you not put your cutoffs in beforehand. You just relax them. And then the system will find the cutoffs for each matrix by itself. It will optimize it automatically. But here we don't have no set. We don't need to optimize any cutoffs. Just run it as it is. So we have to unclick this optimize cutoff option. All right. So let me see how it will work. All right. So by the way, if you have more questions today, probably we don't have more time to answer them, but just send us by email or put it in this form. All right. So you see here, we applied this the whole profile, and it gives us this sort of table summary of how many sites you found for each of that motif in that promoter. And it's all actually calculated in the density of sites. So for the 1,000 nucleotide, we found 33.6, so quite a lot of sites. And if you want to see them, you can select them. Well, let's just see those sites. For instance, you can select several, but I want to see just this one. And here in the output folder, you see also all the sites there, not just this summary. So this is a kind of small representation of those sites. Yeah, we can also click here to see it a bit more in details in the browser. But you see, this is a promoter of that gene of P53, sorry. And in this promoter, we see so many of these HDX sites. You can click on each of that site. And here in the info box, you see which what is the cutoff, what is the score of that site, what is the core score, and what is the consensus of that matrix. And as I said, in the data, when we analyze that, all right, so this folder is quite big. I have many things here, so let me. All right, so you will see all the sites, and actually you can output them. You can also see them in the genome browser. You can output them in your computer. Let me see where they are. Yes, here they are. It creates some kind of a subfolder which have all the information you need. So the summary and also the individual sites. So you see these are the sites, and it found 694 sites. You can open them as a table, so these are your sites. You can also map them on genome browser, and you can export them in different formats. So if you click here on export, you can export them as a BED file, actually, this interval file, or you can export this classical match format, which will give you the text format with all these matrices and position in the genome and the score and on the full score and also the core score. All right, so I think that's it for today. Otherwise, thank you very much, and see you next time. Goodbye.